one slide as an introduction. So Munir Armani's Research Center is a newly established clinical research center that aims to advance science through conducting research in different disease areas. And our goal is to translate our scientific discoveries into effective medical practices for better healthcare. We believe that we share a vision and a purpose that together with an action plan can honor that belief through knowledge, truth, and science to help a better humanity. In today's scientific webinar, we will be talking about immunotherapy since it has been revolutionized the treatment of cancer. Researchers believe that immunotherapy will be less chemotherapy and more targeted approach for essentially all cancer types. So our title for the webinar this month is New Insights and Tools to Tackle Immunotherapy. We have two sessions, and I would like to thank our honorable speakers and guests, Dr. Claude Lambert, and he will talk about the tools to monitor immunotherapy, and Professor Sherine El-Sawa targeting the tumor microenvironment in lymphoma. Dr. Lambert is the charge of the immunomonitoring unit in the Immunology Lab, University Hospital of St Etienne, France. He has involved in cell analysis in immunology and special focus in T-cell receptor B cells development in cancer, immunity, immunodeficiencies, and allergy. Dr. Lambert is a part of international society like the ISAC of European Society and ex-president of the European Society of Cell Analysis. Dr. Lambert is involved in several training initiatives in Europe and North Africa, including the International School of Cell Analysis in Immunology, and that's now held in Geneva University every year, and the Erasmus project in Egypt. I am cert. So Dr. Lambert, in his lecture today, he will explain that the recent emergences of using biologics in treatment with its high cost and unknown side effects, considering the specificity of this type of treatment. And in this talk, Dr. Lambert proposes a different example of monitoring biotherapy by measuring the target and focuses on application biotherapy using monoclonal antibody and precise, quantitative, and cost-effective cytometry application. Thank you for agreeing to be with us today, Dr. Lambert, and please start your talk.
Good evening. Thank you for the nice introduction. It's my pleasure to share this presentation. I'm trying to share my screen. Do you see my screen? And can you hear me?
Yes, very clear.
I'm going to tell you about a few examples of monitoring biotherapy, as it is the theme of this evening. And many working in the hospital, so I shall only show you some practical application of cell analysis in case of in humans. So what is the interest of doing monitoring of biotherapy? First, you have different type of biotherapy. Some biotherapies aim to destroy the targets, like effector cells or tumor cells, for example. And the other type of biotherapy aim to stop, to neutralize some receptor, like adhesion molecules or cytokines. There is many sort of biotherapy available now, and there is more and more coming soon. The condition for this biotherapy to work properly is that you need to have a sufficient quantity of bioavailable biotherapy in the body, and we are looking many in the blood, but it's also important in the tissue. You may have to know about the pharmacodynamic of distribution of your drug after injection and also during the day between two different injections to know and to optimize the frequency of injections and the dose of injections. And also you have to minimize the side effects. And with biotherapy, we do not have much side effects, but we can have, so we have to anticipate it and to try to reduce it. And also biotherapy may, it is less and less the case, but may induce immunization, and so you may, the patient may produce some inhibiting antibodies against this biotherapy. And in some cases, you see that your treatment is not efficient anymore, and you have to know why it's not working. Is it because of immunization? Is it because of a change of target, etc.? So this is important to develop in the setting of a new biotherapy and also in monitoring patients who are treated. So I shall show you some flow cytometry images, and by flow cytometry we can, this is a routine test, it's not very sophisticated. You can easily on peripheral blood identify the lymphocyte, monocyte and granulocyte. From the lymphocyte, you can by focusing on the lymphocyte family and by using a specific immunolabeling. For example, if you use CD3, you can identify T cells. If you use CD56 or CD16, frequently we use the two, you can easily define the NK cells that do not express CD3. And the third population is mainly B cells. And from the CD3 positive T cells, you can easily separate CD4 T cells and CD8 T cells. And you can number these cells in percentage of in concentration. So the first application you can use in biotherapy is to monitor the rituximab. Rituximab is a monoclonal antibody against B cells, which is very cytotoxic. You have a rapid decline of a number of B cells, and as I told you, we can focus on lymphocyte, and then we can analyze the expression of B cells by the presence of B cells by the immunolabeling with CD19, which is an antibody different from the biotherapy. In a normal patient, you can see B cells, which are approximately 20 to 25% of lymphocyte. In patients who are treated with rituximab, you have a disappearance almost complete of circulating B cells. But sometimes the clinicians ask more than this only information. For example, if we follow the maturation of B cells are produced in the bone marrow. They go in the circulating blood, and first at the first step they are transitional cells. Then they will go to the lymph node, and they will complete their maturation to memory cells and plasmablast. And this we can monitor it. For example, we can monitor easily the transitional cells by labeling the B cells with CD38 and IgM, membrane IgM, or it can be also CD24. So you can measure if you still have a production of transitional B cells. We can also measure by labeling IgD, membrane IgD and CD38. You can see that we have different stages of maturation of B cells. This is the early B cells who express IgD and no CD38. Then CD38 appear, and this is the majority of circulating B cells in healthy patients. Then you have still more expression of CD38. before this, you have a class switch, so you lose the expression of IgD, and the B cells will express IgA or IgM or IgE. IgE B cells are very rare. And then you have switch B cells with a decline of the expression of CD38. So you have different steps in the maturation of B cells, and you can see at which step you have a stop in the production of your B cells under the treatment. And most importantly, in the monitoring of biotherapy is to see if you are targeting memory cells that that are responsible of a clinical effect. So the clinicians ask us if we can measure the memory cells, and by plotting the immunolabeling of IgD and CD27, you can clearly identify two populations of B cells, memory B cells. One population still have IgD. This is a marginal zone memory cells that do not enter the germinal center, and they produce IgM. And you have switch memory B cells that do express IgM, IgG or IgA and not IgD, and high level of CD27. And this is the particular target that biotherapy aims to destroy because they include the cytotoxic cells. So in this case, you can see that we have a disappearance of memory cells. We can use also this labeling with for primary to detect the primary immunodeficiencies. And there is also, it can be also be used, but I have no experience in the treatment of antibodies against CD38, which is used in the treatment of myeloma. This antibody specifically target plasma cells. Plasma cells have a switch, so they have IgM, IgG or IgA, and they have very high level of CD38, so it's difficult to see them here, but these cells should disappear, so you can do the same immunolabeling on peripheral blood cells, or even better in the bone marrow. When you have a T cell activation, your T cells will recognize the peptides, it will activate, and the cells will produce first IL-2 receptor, which is you can label it with especially with alpha chains, which is the CD27, 25. This is the first activation molecules that is appearing on the surface of the T cells. And then the T cells will produce IL-2. The IL-2 will have autocrine effect and will auto-activate the same cells. So this is the beginning of the process of T cell activation. And one of the first biotherapy that has been used was the inhibitory of production of and activation of the IL-2. So, for example, in this labeling, again, we have done a different step to separate CD4 and CD8 T cells. And on these two cell population, we measure the expression of CD25. If your cells are not activated, you have a very, very small expression of CD25 on CD4 T cells, which are Tregs, which are very inhibitory T cells. And you have no expression of CD25 on CD8 T cells. If you activate your cells, you induce a good expression of CD25 either on CD4 T cells or on CD8 T cells. And if you have a treatment with anti-CD25, CD25 receptor, then you should limit the activation of your cells. This is the basiliximab that was used in kidney transplantation with little success. So it's not so used now. But they ask us the difficulties of using an CD25 receptor is that you can also inhibit the Tregs, which are supposed to downregulate the immune response. So the clinicians were asking us there is still some Tregs in the patients who were treated with this treatment. And as you can see, we can easily detect CD25 Tregs by using CD127 and CD25. This is an easy way to detect the Tregs. The Tregs do not express CD127, but to express CD25. So you can count the T cell percentage of Tregs among the CD4 T cells. And we could see in this patient that the Tregs have completely disappeared. So this was a bit a worry. I have to. And to check this, we have used the FOXP3. FOXP3 is the gold standard to detect FOXP3, to detect Tregs, but it's a bit complicated to do because you have to do the labeling in several steps and to go inside the nucleus. So this is the CD127 and CD25 is an easy way to do it in practical. But so in this patient, we could see some FOXP3 Tregs. So this we were suspecting an artifact, and we have tested many monoclonal antibodies that were commercially available that target the CD25 for diagnostic, and we could see that most of the monoclonal antibodies that we can use on flow cytometry were completely inhibited by the treatment because the two antibodies are competing on the same epitope of the CD25. So in fact, we were not detecting the Tregs because of competition with the drug. There is only two clones that were not targeting the same epitope, and in this case, you can see this is activated T cells on which we have put basilic the drug, and you could see that in this, with these two monoclonal, you can still see Tregs. Another application on monitoring biotherapy is a similar way. There is a treatment which target an adhesion molecules on activated T cells, and this is alpha integrin, which aim to reduce the production, the migration of activated T cells in cerebral tissue, like in multisclerosis treatment, and you can measure the availability of adhesion molecule on the surface of T cells by doing an immunolabeling with specific antibodies, anti-CD49D, and you can see that this is a study that was done by a colleague in France, and they could show that they have a strong labeling of T cells in patients before treatment, and after treatment, you have a disappearance of the expression of these adhesion molecules. And you can compare the expression of adhesion molecules by in patients with different dosage of treatment, as you can see with G0. It's the pink curve, and you can see the level of expression of molecules is decreasing with the quantity of antibody you are going to use. Then another type of biotherapy is to block cytokines. And there is a lot of antibodies which are available. For example, there is cytokines that do antibodies that block Th2 producing cytokines. So the Th2 cytokines are produced by activated T cells that will induce maturation and immunoglobulins production of B cells, and especially the switch to IgE and allergy, atopic dermatitis, chronic urticaria, etc. So you have biotherapy that block the activity of these cytokines. And in this case, we have done analysis of T cells. Again, it's CD4 T cells and CD8 T cells. If you activate your cells, you can induce the production of cytokines. In this case, it's not related to Th2, but Th1. And you can see that we can detect the activation of the T cells by using CD69, which is an earlier activation marker, but it's the same as CD25. And from these activated cells, you can detect if there is a production of cytokines or not. And this is a way you can use to monitor the inhibition of production of cytokines, especially regulatory cytokines. There is also a lot of application of monoclonal antibody to inhibit cytotoxic factor like TNF-alpha. This is well used largely used in rheumatoid arthritis, for example, and in Crohn's disease. In this case, there is different aspect you have to know. First, after some times, frequently the treatment efficiency is going down. So the clinician is asking us what is the problem. So there is an ELISA test. It's not cytometry, an ELISA test. You can coat your plate with TNF, for example, and you put the serum of the patient. So you can detect if the patient still has some antibody of the treatment in the peripheral blood. If you have some antibody, you wash out the plasma and you can detect the antibody. So you can measure the quantity of residual treatment in the serum. This can tell you that if treatment is going down very fast, you can try to change the treatment, or you can also inject more frequently the drug. But there is different ways this test is not working well. First, if a patient is still producing a lot of TNF, then you will have TNF in the plasma, and the TNF will block the detection of your antibodies to the coated TNF because you have soluble TNF in competition. You also may have difficulties to detect your drug if you have immunization against this drug and antibody that will produced by the patient will bind to the drug and then will compete with your detection system. And most of the time, they also ask us to detect if there is an immunization of this antibody. So you can do another ELISA test on which you coat your plate with the drug, with the antibody. You wash it, you put the serum, and you will detect if you have some antibody from the patient which target the drug. And then you can detect it with a monoclonal antibody, anti-human IgM, IgG, or IgA. Again, you can have some false positive if you have a lot of drug still in the plasma. So your autoantibody will bind to the drug and not to the coated antibody. So this will inhibit the test. There is now an equivalent of this technique that is using chemoluminescence, and it is automated with an instrument ready to use. There is several MARC cofactors that can interfere with the availability of your biotherapy. This is, sorry, my turn. Obesity of the patient will change the pharmacodynamic of your molecules. You can lose also molecules if you have a lot of inflammation, for example, in the mucosae, like in Crohn's disease, because you can have a leaking of your drugs in the area, for example. There is also an effect on hypoalbuminemia. It's not well elucidated, but it's not well understood. But patients with hypoalbuminemia have different dynamic of the drugs in the serum. We have also done an analysis of a recent treatment that target the alpha chain of IL-12 and IL-23. They have a common alpha chain, and these two cytokines will bind to the different receptor, but they have a common receptor for the P40 chain. And there is a biotherapy which is named ustekinumab, which bind to the different P40 of the two cytokines. This will inhibit the activity of these cytokines and strongly reduce the inflammation. And this treatment is very efficient in diseases like psoriasis and rheumatoid arthritis, Crohn's disease, etc. So this directly reduces inflammation in psoriasis, for example. And you have a very nice reduction of disease activity in a very short delay. So they have asked us to monitor the effect, they were looking for the side effect of the drug before it was on the market. And they asked us to measure if there is an effect, and we could see that at time zero, after 12 weeks or 1 year of treatment, you have no change in the number of different subsets of the T cells, including the Tregs and the NK cells. And they also asked us if there was an effect on the cytotoxicity, because this drug was aimed to compete with anti-TNF drug, and they were asking if we have a reduction of anti-tumoral activity, as we can see in treatment with anti-TNF. So we could see that there was no difference by doing a cytotoxic test on tumor cells with NK cells. And there is a paper from the literature who have shown also that there was a reduction of the production of Th17, but and some reduction of Tregs, but this was transient. So in conclusion, you have a lot of needs to monitor biotherapy, and you have a lot of tools by using flow cytometry or a different type of immunodosage. You can analyze the binding of your biotherapy to the target. You can analyze the bioavailability of your biotherapy in the plasma after injection, and they can measure also the efficacy and the side effect. Of course, you have to be very careful about the quality assurance of your test and to have a good repeatability in time. The work I've shown you were mainly done in my lab in the Hospital of St Etienne, which is a small city. But we have done some collaboration with our colleagues in Lyon, with Jean-François Nicolas teams. I just wanted to mention that we have, as it was said in the introduction, we have a collaboration with the Egyptian team on setting up an education program on immunomodulation. And just a map of France to show you where is St Etienne, near Lyon, near Italy, Switzerland, and Paris is here. I thank you very much, you, for your attention.
Thank you very much, Professor Lambert, for this excellent talk. A lot of examples of molecular antibody, how to measure the efficacy and side effects. Thank you. And now Professor Sherine El-Sawa, she's Associate Professor of Immunology at the Department of Molecular, Cellular and Biomedical Sciences, University of New Hampshire. Professor Sherine received her PS in Agriculture from University of Alexandria and Master in Biology from Florida International University and PhD from University of North Carolina at Charlotte. Dr. Sherine studied the initiation of Th1 response to gamma hyperviruses, and for her postdoctoral training, she joined the Mayo Clinic, Minnesota, and studied the role of tumor microenvironment in lymphoma in the Department of Hematology. She continued to study the role of GLI protein and TME and Schultz Center for Developmental Therapeutics. She joined the University of New Hampshire in 2017. Professor El-Sawa will take us on a journey into the tumor microenvironment, beginning with an introduction to the disease model, GLI protein, TME inflammation, the role of TME targeted interleukin 6, and other biomarkers. Dr. Sawa will explain the role of GLI2 in normal and malignant cell, and will discuss the recent ongoing research. Thank you, Doctor, for accepting to be with us today, and we are ready to listen to your talk.
So thank you for the invitation. It's a pleasure to be here. So I'm going to talk about targeting the tumor microenvironment in lymphoma. I'll give you an introduction about the GLI proteins, which are the molecules that we study. We have a very interesting disease model I'll tell you about, and then how we put the information that we get from looking at inflammation, the role of the tumor microenvironment, and some basic immunology projects together to accomplishing our research goal. So the molecules that we study, GLI proteins, these were initially identified as effectors of the hedgehog signaling pathway. This pathway is composed of two receptors, patched and smoothened, and in the absence of ligand, patched normally inhibits smoothened, which is the signal transduction subunit. However, once hedgehog ligand binds, this removes the inhibition that patched has on smoothened, allows smoothened to induce downstream signaling, which ultimately results in translocation of GLI proteins from the cytoplasm in the nucleus where they regulate gene expression. And ultimately, this is going to lead to a biological response, mainly in terms of hedgehog signaling. There are several inhibitors that were designed. So this is a pathway that's important for embryonic development, and everybody has looked at it in a variety of tumors. And several inhibitors for hedgehog pathway, mainly targeting smoothened, were designed. However, these did not produce the clinical responses that were anticipated of this hedgehog pathway. And this is because now we know that in addition, even in the absence of signaling through hedgehog signaling, there are a variety of other pathways, including EGF signaling, TGF-beta signaling, as well as CCL5, CCR3 signaling, which can regulate these GLI proteins. So GLI proteins are a family of transcription factors that have three members, GLI1, GLI2, and GLI3. These members share several biochemical domains. They are known as zinc finger transcription factors because they have these 5 zinc finger DNA binding domains. All of them have a C-terminal transcriptional activation domain. However, only GLI2 and GLI3 have an N-terminal transcriptional repressor domain, which means they can act as either activators or repressors of transcription. Like I mentioned, these are important for hedgehog signaling, so they're important for embryonic development. And in the context of hedgehog signaling, GLI1 and GLI2 are thought of as activators of transcription, whereas GLI3 is known as a repressor of transcription. This is a review that one of my students wrote several years ago, and it shows a variety of pathways, not only hedgehog, but other pathways that have been shown to regulate either the activity of GLI2 or the expression of GLI2. And our lab has focused on GLI2 for the most part for the last decade or so in terms of research. The disease model that we study is a very interesting model. It's called Waldenstrom's macroglobulinemia, which I will refer to as WM for the remainder of the talk. This is a subtype of non-Hodgkin lymphoma. And if we look at this schematic that shows the different stages of B cell development and the different malignancies that can arise from each stage of B cell development, WM sits here between lymphoma and myeloma because the malignant cells show phenotypic characteristics that are similar to both mature B cells and plasma cells. And one of the hallmarks of this particular malignancy is the overproduction of a monoclonal IgM protein that is associated with significant disease symptoms. And in fact, in patients, once IgM levels are high enough, above 3 grams per deciliter, this is the reason to initiate therapy. Otherwise, it's a watch and wait situation. So if we can control these IgM levels, then patients may not need necessarily clinical intervention using chemotherapy. And currently, the only FDA-approved therapy for WM patients is the BTK inhibitor ibrutinib. In the context of the tumor microenvironment, you know, it's a one word to represent a variety of different things that can be happening. There are a variety of acellular components, including extracellular matrix proteins, as well as a variety of cytokines, and cellular components, which includes the immune microenvironment, as well as the stromal and endothelial microenvironment. And because WM is a bone marrow disease, we look at the bone marrow stromal cells as an important component, not only for normal B cell development, but also for malignant B cell development. So we focused on the interaction between bone marrow stromal cells and malignant B cells, and we were trying to understand how cytokines regulate this crosstalk. Ultimately, our goal is to use the knowledge that we gain from all these different disciplines to facilitate the development of novel therapeutic approaches that could actually help patients. So in early work, we found that WM patients have high levels of CCL5 in their serum. And we found that CCL5 didn't really do anything directly to the cancer cells, but it regulated IL-6 levels by bone marrow stromal cells. It did that through signaling through PI3 kinase pathway, which regulates GLI2, and GLI2 in turn regulates IL-6 expression and secretion. And IL-6 has a well-known role in regulating normal and malignant B cell proliferation, as well as immunoglobulin production. And in WM, it induced increases in IgM secretion, as well as cell proliferation. And so in order to utilize the information from that study, there are several avenues we could take. There are several inhibitors that are available in clinical trials, including an oral CCR3 antagonist that targets the CCR3 receptors. There are several first, second, and third generation PI3 kinase and AKT inhibitors. And then there are a couple of different therapies that can target either IL-6 or IL-6 receptor to prevent the crosstalk between the two. So we investigated the role of using the monoclonal antibody tocilizumab, which targets the IL-6 receptor, binds to it, and prevents IL-6 from inducing signaling in the cancer cells. What we did is we co-injected bone marrow stromal cells and WM cells onto the flanks of SCID mice. And then we waited approximately 7 to 10 days for tumors to appear. And then we initiated therapy using tocilizumab. Mice received 100 micrograms of tocilizumab every other day, and then we monitored them for 5 weeks and examined both tumor growth as well as Ig levels at the end point of the experiment. We had 2 groups of mice. One group was injected with one WM cell line, BCWM. The other was injected with a different WM cell line, RPCI, to represent patient heterogeneity. And overall, there was no difference in survival between the 2 groups of mice, regardless of which cell was used. However, when we looked at tumor size, there was a significant reduction in tumor size in mice treated with tocilizumab in the RPCI group. And although we saw a similar pattern in the BCWM group, this was not statistically significant. Interestingly, mice have different sizes of tumors when we inject the different cell lines. So RPCI generates these large tumors, and mice are euthanized because the tumors exceed the allowed size, whereas BCWMs generate these smaller tumors, and the mice are euthanized because they become ulcerated. However, if we take a closer look at the BCWM group, and we look at day 14, which is the last day that we had all animals that were alive, we did see a significant reduction in tumor volume and tumor growth rate. When we looked at IgM levels, so we quantified human IgM levels in mouse sera, we saw a significant reduction in IgM levels in BCWM cells, but not in RPCI cells. And this variability in response may represent the variability that is seen in patients. And interestingly, RPCI cells tend to secrete significantly higher levels of IgM. So if we look at the two groups, there's a threefold increase in IgM levels by this cell line. So perhaps this therapy is more effective in patients who have a little bit lower IgM levels than those with high levels of IgM. We measured mice weights to look at whether or not the treatment was toxic, and really there didn't appear to be any toxicity in the treatment. So we think that it's fairly safe to use. So to summarize this work, blocking the effects of the tumor microenvironment really didn't provide any survival benefit. However, tocilizumab treatment did reduce the relative rate of tumor growth as well as IgM secretion, and therefore using tocilizumab in patients may provide therapeutic benefit, especially for patients with the elevated levels of IgM. And future studies may combine tocilizumab with other treatments, such as either rituximab, which is an anti-CD20 monoclonal antibody, or with ibrutinib, the only FDA-approved therapy for WM, to see whether or not it would provide better efficacy for patients. But we knew that we had shown in early work that if we knock out GLI2 in stromal cells in the tumor microenvironment and then co-inject the mice with stromal cells and WM cells, we see a significant reduction in IgM secretion in mouse serum using two different mouse models, one where we injected both stromal cells and B cells, and another one which we used human fetal bone chip as a model to mimic the tumor microenvironment. So we know that if we target IgM in the, if we knock down GLI2 in the tumor microenvironment, we can target IgM. But we really didn't know whether or not we could target GLI2 therapeutically using an inhibitor. And we wanted to target GLI2, but in order to target GLI2, we first needed to know whether or not GLI2 has any effect on the malignant cells themselves. And so we looked at what GLI2 does to the WM cell biology. So we used a GANT61, which is a Gli antagonist 61, which targets both Gli1 and 2. And when we treat cells with the GANT61, we see a reduction in IgM levels in all three WM cell lines that are available. When we looked for a mechanism of how Gli targeting reduces IgM levels, we looked at receptors for cytokines that are known to target Gli2, to regulate IL-6, IgM, and we found that IL-6 levels were reduced in the presence of Gli inhibition. We confirmed this using qPCR. And then we used two different short hairpin RNAs to target Gli2. We confirm a reduction in Gli2 expression, in IL-6 receptor expression, and a reduction in IgM secretion. And then we knock down Gli2 in WM cells, and we can rescue the effect. We see a reduction in IgM secretion. We can rescue this phenotype by overexpressing IL-6 receptor, and we gain back the IgM levels that we saw because we know that IL-6 signaling regulates IgM secretion. So far, this work we did in WM cell lines, and we needed to confirm this principle in a primary cell model. So we used B1 cells as a primary cell model to confirm some of these work. B1 cells are found in the pleural and peritoneal cavities, and there's a very small population that is present in the spleen. They secrete natural IgM, and it's thought to help protect the body until the B2 cells are activated. They express B220 and they're negative for CD23. And there are elevated numbers of these cells in the BALB/c BYJ mice. So we use them to look at the effect of Gli inhibition in these mice. We took total peritoneal cells and cultured them with or without Gli inhibition using the Gli antagonist. And then we looked at CD23 expression in B220. This is the majority of cells there, which are the B1 cells and a very small population of B2 cells. And we can see that there's a reduction in IL-6 receptor alpha expression in cells treated with the Gli antagonist. And this also resulted in significant reduction in IgM secretion in these cells. So this is something that's not only happening in these WM cell lines, but we think that this is something that is known to regulate IgM. So Gli2 regulates IL-6 receptor alpha, which is known to regulate IgM, and that's how Gli regulates IgM levels in WM cells. But answering this particular question, what Gli2 does to the WM cells, really opened the role for a lot of other questions. So we did this in B1 cells, which is a small population of cells, but we really did not know what Gli2 does to the B2 cells, which are the majority of B cells that we have, and are the normal counterpart for WM cells. So GLI2 deficient mice are embryonically lethal because we know that hedgehog glee is important for embryonic development. So we generated mice with conditional deletion of GLI2 only in B cells using CD19 mediated Cree expression. Overall, wild type and the conditional knockout mice have similar survival, and we can confirm the genotyping, we can confirm expression of the Cree recombinase, the recombined GLI2 allele, as well as the FLOX allele in the different genotypes. These mice have apparently normal bone marrow development of B cells. So we looked at the pre-pro B cells, pro, pre, and immature, as well as mature B cells in the bone marrow, and they appear similar. And we also looked at the different B cell populations in the spleen, and there appears to be similar levels of transitional B cells, as well as the follicular and marginal zone B cells. So it doesn't appear that GLI2 affects B cell development. We were surprised to see that IgM levels were similar in mice serum. However, most of the Ig isotypes were similar, except IgG1 was significantly reduced in the absence of GLI2, but this significant reduction, whether it's biologically significant, we really did not know. So we took out splenocytes from the mice and we stimulated them with LPS to activate the B cells. And again, we expected to see a difference in IgM levels, but IgM levels were similar. However, to our surprise, there were significantly higher levels of total IgG in the absence of GLI2. And when we looked at the different Ig isotypes, we found that IgG1 levels were significantly elevated in the absence of GLI2. We stimulated mice with, we challenged them with LPS, and we found a similar response, although this was not statistically significant. However, perhaps when we increase the number of mice that we use, we would see a statistical significance. So we then purified B cells from the spleens of these mice, and we induced the cells to undergo class switching using a CSR assay. And we found that there was a significant increase in class switching in cells obtained from mice that were deficient in GLI2. And we looked at sex differences in these mice. Most of the differences that we see here were coming from female mice, but there really was no significant difference in IgG1 levels in male mice. So this suggests that GLI2 somehow inhibits class switching to IgG1. So class switching is something that typically happens in normal cells, but in WM, there is no class switching and the cells are stuck at the IgM level. And so we went back to our WM and we looked at GLI inhibition. We knew that when we inhibit GLI, we see a reduction in IgM secretion, but we hadn't looked at expression. And we found that there was a significant reduction in IgM expression when we treat cells with the GLI inhibitor. And this also resulted in elevated transcripts of both IgG2 and IgG4, suggesting that in WM cells, maybe GLI2 is also inhibiting class switching. Interestingly, we also saw an induction in the expression of AICDA, which is the gene that makes AID, which is a protein that is essential for class switching. And absence of this protein in mice results in hyper IgM responses. So in all 3 WM cell lines that we tested, there was a significant induction in AID expression, suggesting that maybe GLI2 is suppressing AID levels in WM cells, and that's why it's blocking class switching in patient samples. And so the question is, where do we go from there? We have a few ongoing directions that we're pursuing in the lab. One is targeting GLI2, because we know that it has an effect on the tumor microenvironment, and now we know that it also has an effect on the malignant cells themselves. And so for this, we're using immunocompromised mice, and we're going to compare the effect of targeting GLI2 in WM cells alone. So we inject the SCID mice with only WM cells and examine GLI inhibition versus control, as well as targeting GLI2 in WM cells in the presence of the tumor microenvironment, so in the presence of bone marrow stromal cells, which are providing support signals for the WM cells. Another goal is looking at really understanding the role of GLI2 in the regulation of the Ig heavy chain, because we know from WM cells and the mice that are deficient in GLI2 that there is evidence that GLI2 is inhibiting class switching. And so with class switching comes a different question. If we inhibit inhibit GLI2 in WM cells to promote class switching, will that promote cell differentiation into plasma cell differentiation and ultimately apoptosis, or will that make the cells essentially become multiple myeloma cells or behave like multiple myeloma cells? And that's something that we don't know. And how does GLI2 inhibit class switching to IgG1? Is it binding to the switch regions or not? We don't know that as well. And then we're also interested in further characterizing the effect of sex as a biological variable, since most of the class switching to IgG1, we see the difference mostly in the female mice, but not as much in the male mice. So I'm going to stop here by acknowledging the folks that are in my lab. Of course, the newer folks are the ones wearing the mask because they joined the lab sometime during the last couple of years. There are a variety of past members that have contributed significantly to this work, our collaborators at UNH as well as at other institutes, and our funding agencies that allow us to do the work that we do. I'm going to leave you with this image of the model that we have so far from the information that we have, and I'd be happy to take questions at the end.
Thank you very much, Dr. Sherine, for this very nice presentation and presenting your research work, which is very interesting. There are a Q&A section, so if anyone has a question, please type your questions, and we have around 7 minutes for answering the questions.
If I may ask Dr. Samira, please.
Oh, sure. Professor
you.
Mohammed Labib is professor of immunology at Tanta University.
Thanks so much. I'm very happy actually with the post. I just came a little bit back. I was at Mansour University. Thank you very much for the two nice presentation. The common questions for both speakers, if they looked at myeloid-derived suppressor cells because they play important role in immunosuppression, and if it under chronic inflammation and also chronic release of certain cytokines like IL-6, and I don't know if you looked at these cells. The other question actually is if anti-GLI or you stated that anti-GLI inhibit class switching in lymphoma, non-Hodgkin lymphoma cell line, but you stated that they don't actually have this phenomena. They don't. So how is it? Does it turn the cells into how to say other phenotype or other kind of differentiate the myeloma or the lymphoma cells into other kind of tumor cells? This was my comment. Thank you very much.
So that's an excellent question. That's the million-dollar question that we're focusing on right now, is if we inhibit GLI2, will that lead to them becoming myeloma cells or myeloma-like cells, or will that induce their differentiation and then basically solve the problem? And we don't know. So this is sort of what we're doing right now, is inducing them. So you could induce their differentiation with CD40 as well as B-cell receptor cross-linking. And then we're doing that plus minus GLI inhibition. That's for the WM cells. And then we want to look at some of the activation markers and memory markers as an indication of whether or not the cells are continuing their differentiation. So that's an excellent question. I don't have the answer. Maybe in 6 months or a year, I'll have answers to that question.
Yeah, it's a very actually interesting observation. And I wonder if you may think about applying the same phenomena using anti-GLI1 in treating cytokine storm or other antibody-based inflammation, like in corona or COVID-19 or whatever. It's a very interesting observation, actually.
So you asked the first question about the myeloid-derived suppressor cells, and we have some data to show that GLI3 regulates inflammation. It regulates inflammation. We've tried this with an atopic dermatitis model. We've tried this in myeloid cells, and we're now looking to see whether or not it plays a role in the M1, M2 sort of differentiation of cells. So it's work in progress. Hopefully, one paper is in revision right now, and hopefully we continue working on that.
Thank you very much.
So apparently there is no any other questions. So now I would like to thank everyone for, especially the speakers, for your time and effort. And it was really our pleasure to have you. And thanks for the audience. Thank you very much, and looking for our next webinar. Have a good night. Bye-bye.
Thank you.
Thank you very much. Good night.
Good night.
Thank you.
Bye-bye.
Good night.
Bye.
Bye-bye.
Bye-bye.